Hello and welcome dear students. Today we are going to discuss about the topic evolutionary genetics. The main objectives of today's lecture are to know the basic concepts of evolutionary genetics, to understand the diversity of life from a genetic perspective, to discuss the use of DNA sequences to study population history and evolution, to know how the hereditary information in DNA controls what an organism looks, to explain how genetics controls the functioning of organisms, to introduce the rapidly advancing field of evolution and its genetic basis, and finally to study evolution of genes, gene families, genetic systems, genomics and populations. Dear students, let's first start with the introduction to evolutionary genetics. Genetics is the study of genes, genetic variations and heredity in organisms and is an important branch in biology because heredity is vital to organisms' evolution. Genetic processes work in combination with an organism's environment and experiences to influence development and behavior, often referred to as nature versus nurture. Evolution, on the other hand, is change in the heritable characteristics of biological populations over successive generations. These characteristics are the expression of genes which are passed on from parents to offsprings during reproduction. Variation tends to exist within any given population as a result of genetic mutation and recombination. Evolution occurs when evolutionary processes such as natural selection, including sexual selection and genetic drift, act on this variation, resulting in certain characteristics becoming more common or rare within a population. Evolutionary genetics describes how evolutionary forces shape biodiversity which is observed in nature. Evolutionary genetics is the study of how genetic variations leads to evolutionary change. It includes topics such as the evolution of genome, structure, the genetic basis of speciation and adaptation, and genetic change in response to selection within populations. Evolutionary genetics is the broad field of studies that resulted from the integration of genetics and Darwinian evolution called the modern synthesis. This field attempts to account for evolution in terms of change in gene and genotype frequencies within populations and the processes that convert the variation with populations into more or less permanent variation between species. Four evolutionary forces, that's mutation, random genetic drift, natural selection and gene flow, acting within and among populations cause microevolutionary change, and these processes are sufficient to account for macroevolutionary patterns which arise in the longer term from the collective actions of these forces. Human evolutionary genetics studies how one human genome differs from another human genome, the evolutionary past that gave rise to the human genome and its current effects. Evolutionary genetics helps us in understanding the differences in the human genome and how evolution has affected development of the genome and acquired diversity, how the organization of the human genome, genetic variation and different tools to analyze genome diversity elucidate the basic concept of population genetics and evolution, and focus on the origins of humans and explicate the genetic basis of the differences between humans and our evolutionary ancestors. Evolutionary genetics is a branch of biology that studies how organisms evolve over time. It is the study of how genetic change results in evolutionary change that is known as evolutionary genetics. It is concerned with the investigation of the evolution of genomic structure. It is the result of the study of population genetics and evolution combined together. Genome evolution is the process through which the size and structure of a genome change over time as a result of environmental factors. Mutation, transduction, and horizontal gene transfer are all mechanisms by which the prokaryotic genome evolves. Dear students, let's now discuss the genome evolution and its mechanism. Gene duplication is a term used to describe the process of copying a gene from one place to another place. The duplication of a specific area of DNA is what this is called. It happens as a result of recombination, aneuploidy, transposition, polyploidy, and a mistake in DNA replication, among other things. Dear students, let's now discuss the elements that can be moved around. A segment of DNA that can be inserted anywhere in the genome is referred to as a transposable element. Consider, for example, the TY elements in Drosophila. The ALU sequence is the most often seen transposable element in humans. The second one is mutation. Accidental mutations are accountable for the alterations that take place in the DNA. As a result of the change in nucleotide sequences, a frameshift mutation occurs which causes the genome to mutate. This is more common in prokaryotes than in other organisms. Exon shuffle is a term used to describe the process of rearranging exons in a genome. Two exons from distinct genes are brought together during this process. This process is responsible for the formation of a new gene. As a result of a new gene is added to the human genome. Dear students, let's now try to understand that do genes change over time? Genes do evolve. That is true. Genes are tiny units or a portion of a DNA molecule that code for proteins and are found throughout the genome. The primary function of a gene is to store information that is necessary for the production of certain proteins required by an organism during development. They also contain the instructions needed to create individual traits like skin, eye, and hair coloration, among other things. Individuals have about 20,000 genes in their genome which are responsible for everything from the structural proteins in our hair to the muscular fibers in our toes and everything in between. The majority of our genes are made up of DNA and are passed down to us from our parents. The evolution of new genes has played an important role in the history of life on the planet Earth. We humans have more genes with a greater variety of functions than any other living species on the planet. The term new gene refers to genes that have arisen from the first time in the relevant evolutionary time scale and have not previously existed. Dear students, let us now understand what is actually human evolutionary genetics in greater detail. Evolutionary genetics is founded on the principle that the genetic record of life is contained in the genomes of living species, and it reveals evolutionary processes and relationships all the way back to the last universal common ancestor of all species together. To find out about this ancestral organism, we have to compare and contrast the most distantly related branch on the tree of life. Comparisons among much more closely related individuals, such as those from the same species, provide evidence on much more recent evolutionary processes. Our ability to read this genetic record has developed enormously in the last few years. Although our confidence that information on our past existence within our heritable material is somewhat older. Genetic evidence comes from two main sources: the genomes of living individuals that must have been passed down from ancestors, and the second, that ancient DNA from well preserved organic remains, which may or may not be represented now in living descendants. Human evolutionary genetics involves the study of how different copies of the human genome differ from one another and from those of our closest relatives, other primates. Differences between genomes also form the basis of anthropological, medical, and forensic genetics. All of these fields or branches of anthropology are experiencing massive advances as a result of two developments. First, the availability of human and non-human genome sequences annotated with important functional elements such as genes and with sites of genetic variation. Second, that the technology allowing analysis of most of this genetic variation across the genome, initially with the development of hybridization microarrays or chips, followed by the development of methods to sequence whole genomes rapidly and cheaply, so-called next generation sequencing. This explosion of information is driving an unprecedented period of innovation that gives us the tools to analyze huge data sets of unparalleled quality and quantity. This wealth of data is itself catalyzing the development of new interpretative methods. In addition, with the publication of genome sequences from other extant primates as well as data from the genomes of extinct members of genus Homo, we can now comprehensively catalog the genetic differences between humans and our closest relatives. These are indeed exciting times for human evolutionary genetics. The genetic record is similarly a complex palimpsest. Variation among modern individuals is shaped by cumulative past processes, which allows us to investigate different times in human prehistory with the same data. Different layers of the past are accessible through the analysis of genetic diversity. Moving from the most ancient to the most recent, we encounter our phylogenetic relationship to other species, the origin of our species, prehistorical migrations, historical migrations, genealogical studies, paternity testing, individual identification. Extracting information on any one past period or event requires careful interpretation to isolate it from previous and subsequent processes. This information can tell us not only about the demographic history and origins of populations, but also something about the environmental challenge faced by those populations through the influence of natural selection on genetic variation. Dear students, let's now understand that a shared evolutionary history underpins our understanding of biology. The great 20th century evolutionary biologist Theodosius Dobzhansky wrote that nothing in biology makes sense except in the light of evolution. So all the sizes, shapes, chemistries, and genes of organisms alive today derive from ancestors that can be traced back over billions of years. All of these features have been shaped by the environmental challenge faced by these organisms and their ancestors. If it were not the case that humans share an ancestor with every other species on the planet, there would be no value in performing any form of comparative analysis. There would be nothing that the Escherichia coli bacterium, brewers, yeast, fruit fly, that is Drosophila, or nematode worm, zebrafish, mouse, or chimpanzee could tell us about ourselves. None of these species is our ancestor, they are our cousins, equally distant in time from our common ancestor. It's our shared evolutionary heritage with these species that makes them such powerful model organisms. To take just one example, sequencing the mouse genome allows us to identify more genes in the human genome than does sequencing the human genome alone. By identifying segments of DNA that are more similar between the two species that could be expected by chance, we can identify regions whose evolution has been constrained by the need to perform a specific function. Some of these regions are genes. In other words, we can identify a gene not because it looks like a gene, nor because an organism treats it like a gene that is makes a product from it, but because it evolves like a gene. Dear students, let's now talk about the big question that what can we learn from our past? From the wealth of data or information on genetic variation among humans and between humans and other apes now at our fingertips, we can address many of the fundamental questions about human origins. For example, we can ask how genetically diverse humans are, when humans split from Neanderthals, or when and where humans originated. We can also address questions about individuals and groups, such as what proportion of genes in African Americans came from Africa, or how genetically similar two humans are. More recently, we have started to address questions about human phenotypes, such as asking which genetic changes make us human or have allowed us to adapt to different environments. All these questions and more are addressed in this module. Some of these new and exciting inferences can be drawn by analyzing the patterns of genetic variation throughout the genome. Others require us to study specific genomic region in detail. With an entire human genome sequence a few mouse clicks away, we can select the most appropriate regions to investigate to address the particular question in hand. There are some fundamental questions about human origins that we cannot currently address, and some that we will never be able to address. What we would like to know about the past and what we are able to know about the past are two separate things. Some questions are unanswerable because of fundamental technical or ethical limitations. We cannot ask, for example, whether two contemporaneous Australopithecine species were infertile, or what combination of mutations would be sufficient to give chimpanzees language skills akin to those of humans. It is important to note that some of the limits to our understanding of the past are not static. Unforeseen technological innovations continue to surprise us. For example, prior to the advent of methods for sequencing ancient DNA, the genetic divergence between Neanderthals and modern humans was unknowable. Now we have a measure to this divergence, not just for one gene, but across the entire genome. Other intuitively attractive questions fail because of mistaken implicit assumptions about the nature of genetic inheritance, such as what was the ancestral biological homeland of population X, or in which country did my ancestors live 1,000 years ago. Given the frequency with which these two questions about individual and population ancestry are asked, it's worth considering why they are unanswerable. To think of a single origin for an entire population is to misconceive how genetic diversity accumulates in a sexually reproducing outbreeding species such as humans. The process of recombination and independent chromosomal assortment divide the genome into segments that have independent genealogical histories. while the ancestry of a single segment of the genome converges on a single common ancestor, each segment has a separate such ancestor. These common ancestors are almost always far older than the population itself, and it is highly unlikely that they were ever present in the same place at the same time. Since the current world population is only 7 billion and past populations were much smaller, such large numbers of ancestors never existed. This is because these potential ancestors were not all different individuals. The same person appears in different places in the family tree. There is another consequence of this reasoning that is worth spelling out, that the potential ancestors of a single individual and world population size intersect within the past 900 years. Before this time, everyone in the world was potentially an ancestor of every living person. Population substructure, the non-random breeding of individuals from different places, complicates this oversimplified calculation, but the essential point remains: the best answer to the question, where did my ancestors live, is everywhere. Dear students, molecular biology and computational biology offer a tool to understand the evolutionary genetics in a better way. The databases and softwares are going to increase our understanding of finding the unanswered questions in evolutionary genetics. Molecular evolutionary genetics analysis, or MEGA, is one such tool. Dear students, let's now understand MEGA, that's molecular evolutionary genetic analysis. Molecular evolutionary genetics analysis, or MEGA, is computer software for conducting statistical analysis of molecular evolution and for constructing phylogenetic trees. It includes many sophisticated methods and tools for phylogenomics and phylomedicine. It is licensed as proprietary freeware. The project for developing this software was initiated by the leadership of Masatoshi Nii in his laboratory at the Pennsylvania State University in collaboration with his graduate student Sudhir Kumar and postdoctoral fellow Koichiro Tamura. Comparative analysis of molecular sequences data is essential for reconstructing the evolutionary histories of species and inferring the nature and extent of selective forces shaping the evolution of genes and species. MEGA is a user-friendly software for mining online databases, building sequence alignments and phylogenetic trees, and using methods of evolutionary bioinformatics in basic biology, biomedicine, and evolution. The molecular evolutionary genetic analysis, or MEGA software, was developed with the goal of providing a biologist-centric integrated suite of tools for statistical analysis of DNA and protein sequence data from an evolutionary standpoint. Over the years, it has grown to include tools for sequence alignment, phylogenetic tree reconstruction and visualization, testing an array of evolutionary hypotheses, estimating sequence divergences, web-based acquisition of sequence data, and expert systems to generate natural language descriptions of the analysis method and data chosen by the user. Dear students, at the end of the lecture, let's give a timeline of the important events in the history of human evolutionary genetics. This timeline is represented in the form of tables from 17th century to the 21st century. These timeline mentions about the important discoveries, events, and innovations and new technologies which are relevant with the evolutionary genetics from 17th to 21st centuries. Recently, in 2017, single nucleotide polymorphism database, that is DBSNP, which lists SNP and other variants, listed a total of 324 million variants found in sequenced human genomes. Recently, in 2019, scientists discovered evidence based on genetic studies using artificial intelligence that suggested the existence of an unknown human ancestor species, not Neanderthal, Denisovan, or human hybrid like Denny hybrid homin, in the genome of modern humans. Dear students, let's now conclude the lecture. Evolutionary genetics uses patterns of genetic differences between individuals and between species to make inferences about the past. New technologies are providing vast amounts of data, and new analytical methods are being developed to infer evolutionary processes from these data. Human evolutionary genetics can help to explain the distribution and frequencies of different phenotypes and diseases in humans today. The information on human evolution from genetic studies, placed in the context of information from other scientific disciplines, including archaeology and linguistics, provides a synthetic view on our past. Asking clear, answerable questions is an important aspect of studies in human evolutionary genetics. Human evolutionary genetics seems likely to undergo a revolution over the next few years as a tsunami of genetic diversity data flows from new sequencing and genotyping platforms. These data will primarily come not from academic labs but from the clinic and from consumer-driven testing. Similarly, the interpretation of such data seems likely to shift away from academia toward people who are not professional scientists analyzing publicly available genetic data sets, often in more detail than the professionals can. Dear students, in a nutshell, we can say that citizen science is on the rise in terms of evolutionary genetics. Dear students, it was all about today's lecture regarding evolutionary genetics. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.